Transneuronal propagation of pathologic alpha-synuclein from the gut to the brain models Parkinson's disease. This study was done by the following people listed below. It was published in Neuron on August 21st, 2019. The purpose of this study was to develop a model that accounts for the potential spread of pathologic alpha-synuclein measured by phosphorylated on serine 129 alpha-synuclein from the gut to the brain, per Brock and his colleagues' predictions in previous work. The model developed was to inject preformed fibrils into the muscularis layer of both the pylorus and duodenum and to assess the spread of alpha-synuclein commonly observed in Parkinson's disease. Most of the common neurodegenerative diseases, including dementia and Parkinson's, are grouped based on aggregates of the protein alpha-synuclein, and as a group are known as synucleinopathies. In regards to dementia and this protein, it is the aftermath of a loss of connections in cognitive circuits, commonly through cell death. Previous studies by Brock et al. Has pointed out that, quote, neuronal connectivity within this circuit may underlie the path of disease progression and circuit dysfunction associated with Parkinson's disease and potentially other synucleinopathies, including dementia with Lewy bodies, end quote. Brock and his team hypothesized that alpha-synuclein pathology can spread in the gastrointestinal tract from the vagus nerve to the ventral midbrain, where it kills dopamine neurons of the substantia nigra pars compacta. It should be noted that this pathology is not at the beginning of disease progression, but rather thought to be towards the middle. Other studies indicate that fibril forms of alpha-synuclein are transmitted from cell to cell in a prion-like manner, meaning that the protein goes through some conformational change and then spreads in its accumulation. Other studies also indicate that the majority of these alpha-synuclein in fibrils is phosphorylated on serine 129. The mechanisms are unclear, and it is not understood if all neurons that have this phosphorylated serine will transform into Lewis bodies, that is, protein aggregation. Other studies have also shown that alpha-synuclein can form fibrils in vitro to mimic those in vivo. They are known as preformed fibrils. Previous research has shown that these preformed fibrils injected into the striatum have resulted in cell death of substantia nigra pars compacta dopamine neurons, but Brock's hypothesis has not yet been tested of gut-to-brain transmission. Therefore, we want to test a model of this gut-to-brain transmission of pathologic alpha-synuclein and hypothesize that injection of preformed fibrils into the muscularis layer of the pylorus and duodenum should result in immunoreactivity spreading in the dorsal motor nucleus of the vagus nerve, caudal portions of the hindbrain, the locus coeruleus and basolateral amygdala, the dorsal raphe nucleus, and finally the substantia nigra pars compacta. We also hypothesize that truncal vagotomy will prevent this spread from gut to brain from occurring, as well as prevention in alpha-synuclein knockout mice. Alpha-synuclein pathology spreads from the enteric nervous system to the central nervous system through the vagal nerve. To test this statement, mouse alpha-synuclein preformed fibrils were injected into the muscle layers of the pylorus and duodenum primarily because these two areas are innervated by the vagus nerve. Quote, alpha-synuclein preformed fibrils was prepared in PBS of alpha-synuclein monomer by stirring with magnetic stir for 1 week. Alpha-synuclein aggregates were sonicated for 30 seconds, and to determine the length of alpha-synuclein preformed fibrils, alpha-synuclein preformed fibrils after sonification were imaged by transmission electron microscopy, end quote. 2.5 microliters of preformed fibrils was injected into 2 different locations in both the pylorus and duodenum because 3 microliters was determined to be the maximum safe injection amount without causing leakage or damage. The mice injected were 3 months old. Sangjun says, quote, the myenteric plexus of the pylorus and duodenum were examined and demonstrate phosphorylated on serine 129 alpha-synuclein immunostating in myenteric neurons, indicating that the preformed fibrils are templating the endogenous alpha-synuclein to misfold. Also, phosphorylated on serine 129 alpha-synuclein immunostaining was used to display the progression of alpha-synuclein from the gut to the brain, end quote. It was found that a month following the injection, the phosphorylated serine was seen in the dorsal motor nucleus of the vagus nerve and the locus coeruleus. 3 months in, there is accumulation in the amygdala and substantia nigra pars compacta, of the ventral midbrain, as well as small amounts in the hypothalamus and prefrontal cortex. 7 months in, it is still in all the above-mentioned areas, as well as the hippocampus, olfactory bulb, and striatum. Finally, we observed after 10 months that the phosphorylated serine was increasing in the hippocampus, prefrontal cortex, olfactory bulb, and striatum, but we found that at this time point the levels decreased in the amygdala, ventral midbrain, and the medulla oblongata. Using the same injection procedure, we also followed dopamine neurons' life after injection of preformed fibrils into the gut. Tyrosine hydroxylase and Nissl-positive cell counts were analyzed by using serologic methods. To measure tyrosine hydroxylase and Nissl-positive cell counts, we used immunohistochemistry techniques. Brains were collected and fixed for 16 hours, coronally sliced, and then we used an antibody against tyrosine hydroxylase and also phosphorylated serine 129. Quote, after developing stains using DAD peroxidase, sections were counterstained with Nissl. Tyrosine hydroxylase positive and Nissl positive dopamine neurons from the substantia nigra pars compacta region were counted through optical fractionators, the unbiased method for cell counting, by using a computer-assisted image analysis system. End quote. After both 1 and 3 months after injection, there was no significant loss in these cells. However, by 7 months, there was a significant loss of dopamine neurons in the substantia nigra pars compacta, and the loss got greater and greater at 10 months. In addition to the dopamine neuron loss, there was a decrease in tyrosine hydroxylase immunoreactivity in the striatum. We also utilized high-performance liquid chromatography to discover that striatal dopamine is decreased at 3 months and even further decreased at 7 months post-injection. Finally, we were able to determine that multiple dopamine metabolites were also reduced by 7 months. The question for this area of our experiment was to determine if pathologic alpha-synuclein is indeed sent from the gut through the vagus nerve, and if endogenous alpha-synuclein is necessary for this distribution to higher brain regions. To do this, we did experiments after performing a truncal vagotomy after preformed fibril injection into both wild-type mice or alpha-synuclein knockout mice. Quote, after the intestinal alpha-syn injections, a full truncal vagotomy was performed. The procedure is as follows. The vagus nerve was identified where it exits, the diaphragm and the gastric branches were identified. From 2 to 3 millimeters above the cardia, a 4 to 6 millimeter section of each gastric branch was carefully isolated from the surrounding tissue and overlying vasculature. The neck incision was closed with black silk. The abdominal incision was closed with sutures, and the skin incision was closed with wing clips. End quote. To determine the pathology, we analyzed these injected mice 7 months later and found that there is a significant increase of phosphorylated on serine 129 alpha-synuclein in the duodenum of wild-type and truncal vagotomy mice, but not the knockout mice. When looking at the substantia nigra pars compacta, we found that 7 months post-injection, phosphorylated on serine 129 alpha-synuclein is in the wild-type mice, but not the truncal vagotomy or knockout mice. We also found preformed fibril injection results in a loss of dopamine neurons, which was found to be prevented by truncal vagotomy or in the knockout. Overall, it's clear that alpha-synuclein can spread from the GI tract to the brain via the vagus nerve. Since it is widely accepted that pathological alpha-synuclein is associated with Parkinson's disease, we can now begin to include the enteric nervous system in its description. It also has implications because ENS detections occur prior to CNS detections, and prior studies showed that this has to do with, quote, increased intestinal permeability, which results in oxidative stress, end quote. Other studies have also shown that curly, synthesized by the microbiome, may have implications in alpha-synuclein aggregation and motor dysfunction. It was also found that enteroendocrine cells in the gut might be a location at which toxins first encounter the alpha-synuclein prior to the pathogenesis to the brain. Our findings support Brock's hypothesis in that Parkinson's disease may begin in the GI tract, spread via the vagus nerve to the brain by means of phosphorylated serine 129 alpha-synuclein through brain regions like the dorsal motor nucleus of the vagus nerve. This was verified in the aforementioned experiment of the truncal vagotomy because it resulted in the phosphorylated on serine 129 alpha-synuclein not being present in the SNPC. All of the locations we found the alpha-syn in the brain spoken of in the results correspond to different stages of Parkinson's that Brock hypothesized about, and thus provide even stronger support for the hypothesis. We also found that dopamine neuron death in the SNPC and decrease in TH in the striatum. One point of concern when comparing these findings with knowledge already known is that the olfactory system was not affected until months later in our model than other models. Parkinson's is commonly thought to have early involvement of olfactory system, but only the GI tract had early involvement for our studies. The only critique of this study is the lack of foresight on their part. They should have looked out further than 10 months to ensure there was nothing else happening.